Assalamu alaikum dear students. Welcome to Dr. Roomi lectures. In today's session, we will study the mechanism of action of insulin and insulin's actions on carbohydrate, lipids and protein metabolism. Insulin is a major anabolic hormone which is created in response to carbohydrates and proteins. Anabolic hormone is that hormone which increases protein synthesis in our body. And some examples of anabolic hormones other than insulin are thyroid hormones, growth hormone, insulin-like growth factor 1, also called as somatomedins, and sex steroids, especially androgens. In order to understand the mechanism of action of insulin, let's talk about insulin receptor. Insulin receptor is present in our cell membrane because insulin is a water-soluble hormone, and water-soluble hormones have their receptors in the cell membrane. This receptor is a tetramer, which means it's made by 4 polypeptides. There are 2 smaller units called as alpha units which are present on the outer surface of the cell membrane, and 2 beta subunits which are passing through the cell membrane. Insulin binds with the alpha subunit on the outer surface of the cell membrane, and because beta subunit is linked with the alpha subunit, so whenever alpha subunit is occupied by insulin, beta subunit, which is having tyrosine kinase activity on the inner side, will be stimulated, and then it causes phosphorylation of the beta subunits, which is called as autophosphorylation. And not only it causes phosphorylation of itself, but it also causes phosphorylation of some other proteins, most of which are enzymes. So many enzymes are phosphorylated by the tyrosine kinase activity of the beta subunits of insulin. A group of enzymes called as IRS or insulin receptor substrate is also activated. These enzymes, which are phosphorylated, affect the protein, fat and carbohydrate metabolism. By this phosphorylation process, some enzymes are activated and some enzymes are inhibited. There are different types of insulin receptor substrates, IRS1, IRS2, IRS3, and different cells express different types of insulin receptor substrates. So if someone asks you what is the mechanism of action of insulin hormone, you should say because it's a water soluble hormone, it will have its receptors in the cell membrane. And secondly, its mechanism of action is enzyme linked receptor. Very specifically, we call it tyrosine kinase activity because the inner part of the beta subunits is having tyrosine kinase activity. And as you know that kinases are those enzymes which cause phosphorylation. Not only the beta subunit is phosphorylated, but also a group of enzymes inside the cytoplasm, they are phosphorylated. And by phosphorylation, some enzymes are activated and some enzymes are inhibited. And in this way, insulin directs the cellular metabolism of carbohydrates, lipids, and proteins in the desired direction. A very important clinical point to note is that insulin downregulates its own receptors. So when we have too much activity of insulin in our body, as happens in case of obesity, because more insulin is produced in obese people, then it will decrease the number of its own receptors and then the insulin resistance may develop. And in those cases when we are starved, because there is less insulin production in our body, then the insulin receptor number will increase. So it will increase the number of insulin receptors when there is less insulin being produced in our body. Now let's see what are the short-term, intermediate term and long-term actions of insulin. So when insulin acts on its receptors, very quickly within seconds, there is increase in uptake of glucose. This is by diffusion process. And this happens because the membrane vesicles which contain the carriers for glucose diffusion called as GLUT4 carriers, these vesicles are translocated back to the cell membrane, and hence the number of GLUT4 carriers in the cell membrane will increase and it will cause diffusion of glucose into the cells of adipose tissue and resting skeletal muscle. So this GLUT4 transported in adipose tissue and skeletal muscle is dependent on insulin, and through this transporter there is diffusion which is not linked with the sodium. Very interestingly, when insulin is not available, then these GLUT4 transporters will be internalized through the membrane bound vesicle into the cytoplasm of the cell, and this happens very quickly within 5 to 10 minutes. Although resting skeletal muscles need insulin for the entry of glucose into the cells, however, working skeletal muscles or the contracting skeletal muscles, do not need insulin for the entry of glucose into the cells, which means that whenever we do physical activity, there is more entry of glucose into our cells, and for this we do not need insulin. It's because the contracting skeletal muscle can bring these GLUT4 transporters from inside the cytoplasm to the cell membrane without requiring insulin. This is why those who are having diabetes, initially some important strategies to control their diabetes are to increase physical activity, and secondly to decrease their weight, because by decreasing weight there will be less adipose tissue in the body, and because when there will be less adipose tissue there will be less need for insulin, because adipose tissue needs insulin for the entry of glucose into the cells. And also when we do activity or exercise, once again without the presence of insulin, these GLUT4 transporters can be translocated into the cell membranes. Do you know that there are tissues in our body which do not need insulin for the entry of glucose into the cells? And these tissues are nervous tissue, tubules of kidney, intestinal mucosa, red blood cells, and beta cells of pancreas. Just to link it with renal physiology and membrane transport, the carrier which is required for transport of glucose in kidney and intestines is different one, and that uses sodium for secondary active co-transport of glucose. So that's why they do not use insulin. The carrier here is GLUT4 transporter which uses insulin for the entry of glucose into the cell. What about liver? Insulin accelerates but is not required for the uptake of glucose by the hepatic cells. And insulin does this in the liver cells by increasing the metabolism like glycolysis and Krebs cycle, but not by increasing the GLUT4 transporters in the cell membrane of liver cells. Insulin also increases the permeability of the cell membrane to potassium ions and also amino acids, so their entry into the cell is increased. After the action of insulin on its receptors, within next 10 to 15 minutes, there is increase or decrease in the activity of some enzymes into the cell, and this is because of phosphorylation, and this affects the carbohydrate, lipid and protein metabolism. Insulin also performs some action slowly over hours to several days, and this is because of change in the rates of transcription of DNA to make messenger RNA, and also change in the rate of translation of messenger RNA to make different proteins. So this activity of transcription and translation for some genes is increased and for some genes it is decreased. Now let's list the actions of insulin on carbohydrate metabolism. The first action of insulin is to increase the entry of glucose into the cells. Let's suppose we are talking about fat cells and the muscle cells. When glucose entry into the cells is increased, then first thing is glucose is trapped into the cell by its phosphorylation. This is because insulin increases the activity of glucokinase enzyme, especially in the liver. When glucose 6-phosphate is formed inside the cells, then there is increase in the activity of phosphofructokinase by the action of insulin, and this increases the rate of glycolysis, and also the oxidation of glucose in the Krebs cycle is increased. So after taking a meal, when the blood glucose level rises, there is production of insulin. That insulin increases the entry of glucose into the cell. Glucose is oxidized. Some glucose is also stored in the form of glycogen. In this way, the glucose level calms down. What do you think? What is the favorite fuel of a cell 4 to 5 hours after a meal when the glucose has already been used? The answer is fatty acids. The resting skeletal muscles, they prefer fatty acids when we are in a hungry state or fasting state. But when we take a meal, because there is excess of glucose, then our resting skeletal muscles, they use glucose, and this is in the presence of insulin. Insulin also increases the conversion of glucose into glycogen in liver and also in skeletal muscles. This is by increasing the activity of glucokinase enzyme and glycogen synthetase enzyme. Please keep in mind, glucokinase enzyme is insulin dependent and that's present in liver. The other kind of enzyme which converts glucose into glucose 6-phosphate is hexokinase. It's not insulin dependent. So what happens with glucose after taking a meal? It depends how much carbohydrate you have ingested. If you have taken just a little bit of carbohydrate, then it will be enough to just provide you energy. But if you have taken more carbohydrate, then the extra glucose will be stored in the form of glycogen. And this glycogen is stored in muscle and liver. The glycogen which is stored in liver is generous, because when we are fasting, then liver can break that glycogen and give it to our blood so that glucose can be provided to our other tissues. But this is in the absence of insulin in case of fasting. What is the importance of glycogen which has been stored in our muscles? This is important to give energy to our muscles, and this glycogen is especially important for glycolysis. You see, glycolysis can provide us ATP at a faster speed than aerobic respiration, and this can be enough to give us ATP at a faster speed for a few minutes. For example, if you are running a 100-meter race, this may take like 1 to 1.5 minutes or 2 minutes, and in such a case of rapid supply of energy, this glycogen will be a good source in our muscles. Insulin not only increases the synthesis of glycogen, it also decreases the glycogen breakdown. So glycogenolysis is decreased both in liver and also in muscle, and this is by decreasing the activity of two important enzymes. One is glycogen phosphorylase and the other one is glucose 6 phosphatase. So what will happen when we will be fasting or when we will be hungry? In such a case, insulin level will be low. Then the activity of these two enzymes, glycogen phosphorylase and glucose 6 phosphatase, in the liver will be increased. And because glucose 6 phosphatase is present only in liver, so that will increase the release of glucose into the blood. So liver is generous. In case of fasting conditions, it will provide us glucose which will be coming from the glycogen breakdown, and this is only when insulin level will be low in fasting case. Insulin also decreases gluconeogenesis in liver. This is because we do not want any more glucose. We already have enough glucose in our blood after a meal, and that glucose must go to other cells. It must be used, it must be stored in the form of glycogen. So we don't need any more glucose. So the new synthesis of glucose, that is gluconeogenesis, it will be decreased in our liver, and this is possible by inhibiting the enzymes of gluconeogenesis. The most important rate limiting enzyme of gluconeogenesis is phosphoenol pyruvate carboxykinase, which is inhibited by the action of insulin, and that's why gluconeogenesis or new synthesis of glucose in the liver will be suppressed. Now, as I have mentioned, when we take a little bit of carbohydrate like just one biscuit, so most of that carbohydrate will be used as a source of energy in glycolysis in Krebs cycle. When you take a little bit more carbohydrate, the extra carbohydrate will be stored in the form of glycogen. But what if you take a lot of rice or roti, a lot of extra carbohydrates? What will happen? These extra carbohydrates will make a lot of glucose for you, and these extra glucose molecules will be converted into fatty acids and will be stored in your fatty cells in the form of triglycerides. We have limit for the storage of glycogen, but we don't have any limit for the storage of triglycerides. Do you know the maximum capacity of our liver to store glycogen is just 100 grams? Just 100 grams. But how much is the capacity of our body to store lipids? Unlimited capacity. We can store kilos and kilos of fats in our body. So this is why if you eat a lot and not doing any physical activity, the extra form of carbohydrate will be converted to fats and will be stored in the adipose tissue in the form of triglycerides. Now let's talk about the actions of insulin on protein metabolism. Insulin increases the entry of amino acids into the cells, and these amino acids, when they enter into our cells, the blood level of amino acids will be decreased. What will happen when there is deficiency of insulin in our body, as in type 1 diabetes, or when there is less action of insulin, as in type 2 diabetes? The level of amino acids in our blood will increase because they are not entering into the cells. Now, in the presence of insulin, when the amino acids have entered into the cells, there is increase in the synthesis of proteins. So insulin increases protein synthesis. At the same time, not only it increases protein synthesis, it also decreases the protein breakdown. In this way, because the breakdown of protein is decreased, so that's why there will be less urea formation, or the ureagenesis will be decreased. In case of diabetes mellitus, when there is less action of insulin, there will be more protein breakdown, and the people with poorly controlled diabetes will have protein breakdown, and gradually the size of their muscles and their bulk will decrease. Now let's talk about the actions of insulin on fat metabolism. Insulin increases the entry of glucose into the liver cells, and this glucose in the liver cells, if it is extra, then it is converted into fatty acids. This is called as de novo synthesis of fatty acid. The rate limiting step in the synthesis of fatty acids is the conversion of acetyl coenzyme A into the malonyl coenzyme A. The enzyme for this reaction is activated by the presence of insulin. Hence, there will be more synthesis of fatty acids, and there will be increase in triglyceride formation in our liver. So that VLDL, which is a kind of lipoprotein made in our liver, will be sent through blood to our adipose tissue. In the adipose tissue, there is an enzyme called as lipoprotein lipase enzyme that acts on the VLDL. The triglycerides are broken down, and then the fatty acids will be given to our adipose cells. Those fatty acids inside the adipose cells, they need glycerol 3-phosphate, so that the glycerol and the fatty acids, they can be joined together to make triglycerides. Now the glycerol 3-phosphate in our adipose cells will be made from the glucose which has already entered into the cells in the presence of insulin. So we can say that insulin increases lipogenesis, and at the same time it decreases lipolysis. So the breakdown of lipids will be decreased, but the synthesis of lipids, it will be increased by the action of insulin. Now what will happen when insulin is not present in our body, and when there is decreased action of insulin, as in case of diabetes mellitus? There will be breakdown of lipids, and those lipids are converted into fatty acids, and then fatty acids in our liver are converted into ketone bodies or keto acids like beta hydroxy butyric acid. So this will lead to acidosis in case of uncontrolled diabetes mellitus. Especially in type 1 diabetes mellitus, there is development of an acidosis which is called as DKA, diabetic ketoacidosis, because of too much formation of ketone bodies. This is a deadly condition and hospital emergency. If it is not treated, this can cause death of the child. Insulin not only affects the metabolism of carbohydrate, lipids and proteins, but it also affects a very important electrolyte which is potassium. It increases the activity of sodium potassium ATPase pumps in most of the cells, and hence it increases the entry of potassium ions into the cells, thus decreasing the blood levels of potassium ions. This action of insulin on causing the entry of potassium ions into the cell has two important implications. First, in case of uncontrolled diabetes mellitus, because insulin is not available or it's not acting, so the entry of potassium into the cells is decreased. And secondly, in case of diabetic ketoacidosis, as there is acidosis in the body, in order to buffer the hydrogen ions from the extracellular fluid enter into the cells and potassium ions comes out. This also increases the level of hyperkalemia. And in this way, in case of uncontrolled diabetes mellitus, due to decrease in the action of insulin and also because of acidosis, the life-threatening hyperkalemia may develop. In another case of chronic renal failure, where there are also chances of increase in the level of potassium ions, because kidney is a very important ion which regulates your potassium ion level. So when there is kidney failure, the excretion of potassium from the kidney will decrease and the blood potassium level will increase, and sometimes this can become life-threatening. How do we treat it? So when potassium level is very high, we can treat it by giving insulin to the patient, and insulin is given along with glucose. Insulin will send potassium into the cell, in this way treating the hyperkalemia. And why do we give glucose along with insulin? Because if you give insulin alone, this can cause hypoglycemia in the patient. We give glucose so that insulin induced hypoglycemia does not occur. So just to summarize, when you have insulin, it will sell potassium into the cell, and when there is no insulin, then the potassium level will increase in our blood plasma. And when we have high potassium level in our plasma, we can decrease it by giving insulin along with glucose, as in case of chronic renal failure. Now just to summarize, insulin is a protein hormone which acts on a receptor which is present in the cell membrane, and this receptor uses tyrosine kinase mechanism. It's an enzyme linked receptor. After this receptor is activated, there is phosphorylation of several enzymes inside the cell, and because of that phosphorylation, some enzymes are activated and some enzymes are inhibited. Very quickly after this activation of the receptor, the GLUT4 transporter is translocated from inside of the cell using the vesicle into the cell membrane, and this increases the entry of glucose into the skeletal muscle and adipose tissue. Please keep in mind, adipose tissue and skeletal muscle are two insulin dependent tissues where diffusion of glucose needs the requirement of insulin. Insulin is an anabolic hormone, and this affects the carbohydrate, lipid and protein metabolism. It increases the entry of glucose, amino acids, fatty acids and potassium ions into the cells. Insulin promotes use of glucose for oxidation through glycolysis and Krebs cycle. It also increases the storage of glucose in the form of glycogenesis, and if we have taken more carbohydrate, then the extra glucose will be stored in the form of fats. So it increases lipogenesis. Insulin also increases protein synthesis, and at the same time insulin decreases lipid breakdown, protein breakdown. Insulin also decreases the breakdown of glycogen or glycogenolysis. It also decreases gluconeogenesis or formation of new glucose. It also decreases the formation of ketone bodies or ketogenesis. It also decreases ureagenesis because the breakdown of proteins into amino acid and then the amino acid metabolism to make urea is decreased. So insulin is the most important hormone which decreases our blood glucose level after a meal by sending it into the cells, storing it, and by not allowing the formation of new glucose. There are several other hormones in our body which tend to increase blood glucose level, and we call them anti-insulin hormones or diabetogenic hormones. Some examples of anti-insulin hormones are cortisol, glucagon, growth hormone, epinephrine, and thyroid hormones. I hope you must have enjoyed this session in which we have covered the physiology and biochemistry of insulin. In our next session, we will study the types of diabetes and its clinical features. Thank you so much for watching this video. See you next time with another video.